We're gonna go ahead and get started. Thank you all for joining us. We're excited to be here today. First off, this is exciting because actually we're sponsoring our STEM education seminar today from 3 different departments in the Center for STEM Teaching and Learning, Department of Biological Sciences, and the Department of Philosophy. And so we're excited to have such an interdisciplinary talk here today. And I think Dr. Fagan's talk kind of hits on all 3 of those areas, which is why we thought this would be so perfect. All right, here's the part where I read things. So first off, Dr. Melinda Fagan is an associate professor of philosophy at the University of Utah, where she holds the Sterling M. McMurrin Chair. Dr. Fagan is a leading scholar in the philosophy of science with a particular focus on experimental practice in biology, explanation of the nature of scientific knowledge. She holds 2 PhDs. That's quite impressive. Some people here have none. I'm just saying. She holds 2 PhDs, one in biology from Stanford University and another in the history and philosophy of science from Indiana University. And her interdisciplinary background makes her uniquely positioned to engage our 3 audiences today. For those of you joining us on Zoom, hello. For those joining on Zoom, I'll be monitoring the chat. And so at the end, when we have time for questions, I'll be sure to read yours off. We ask that you stay here because that's the only camera we have, so she's not gonna get to walk around too much. But with that, will you join me in welcoming our speaker today, Dr. Melinda Fagan.
Oh, thanks so much, Ron. Oh, the Zoom is seeing the presenter view. I don't know what that means.
Oh, yes.
But maybe that's good. So thanks so much, Ron and everyone, for coming out. I hope folks can hear me all right, both in person and on Zoom. This talk, long kind of involved title. The talk is going to chart a path from biology to philosophy. Models of development, models of knowledge. The development is biological development. The models of knowledge is going to be central to a lot of philosophical studies, but I hope that there's going to be some broader issues and implications that come out toward the end. So here's the plan of the talk. I'm aiming for it to be about an hour. I'm going to try to keep an eye on the time. I'm going to start out with some important experimental models in stem cell biology. These are organoids and embryo models. Those are the living experiments of the title. And I'm going to start with their scientific role and just walk through what they are. And then I'm going to contrast that role, moving to point 2 here on the outline, to the main philosophical theories of scientific models and modeling. And I see the relation between science and philosophy, like most productive interdisciplinary relations, as a 2-way street. You can get insights from philosophy, helping to clarify some big picture issues in science, organoid research, for example. And in earlier work, I hope that's not too blurry. In earlier work, I've done that. I've looked at ideas from philosophy of science and kind of applied those directly to organoid research as a way of getting insights that can complement what's going on in the science. What I'm going to do in this talk is work more in the other direction to look at living models like organoids and embryo-like structures, and then from there extract some insights for philosophy of science. But I do think then that those insights can feed back and inform broader ideas about science and society, which will be of interest to philosophers, scientists, and a broader audience. The main idea that I'm going to argue for in this talk is this point number 3, the thesis, such as it is that knowledge in fields like stem cell biology, and maybe in many other areas, take the form of an intricate fabric connecting models with one another and with their targets. And in the last part of the talk, I want to extend that idea further to look at science more broadly in that image, and then consider some of the implications of that idea. That's point number 4. So that's the overall plan of the talk. I'm going to start looking closely at some important biological models, and from there build outward to more general claims and ideas about scientific knowledge. So here we go. The models of development. These are the first part of that title. These are organoids and embryo models. They're constructed using stem cells to model, it's right there in the first little blurb, human development and disease in novel ways. I'll get to what's novel here shortly, but the point I want to start with is that both these kinds of models are made from stem cells, and more precisely from cultured stem cell lines, which are themselves a kind of experimental model. the theme of the talk, there are a lot of models here to untangle. So I'm going to back up and look at the origin of both these kinds of models, stem cells. I imagine that that term is familiar to most folks. Stem cells are defined in general as cells that have two abilities. They can reproduce by cell division and make more stem cells of the same kind, just make more like themselves. And they can also give rise to more differentiated cells. That is, more specialized cells that are characteristic of later developmental stages. There's kind of an implicit idea or theory of development in the very notion of a stem cell, that it is relatively unspecialized and undifferentiated, lacking whatever features make a cell functional in an organism. So those abilities in stem cell biology are called self-renewal, the reproduction by cell division, and differentiation, the ability to give rise to other kinds of cell that are more specialized, that are further along in the developmental process. And right there, that's actually kind of unusual for a cell in biology and medicine, because most of the time cells are defined, if you have a type of cell, it's defined by features that you could observe under a microscope or just examine looking at the cell itself. But stem cells are defined functionally, not by features that they have, but things that they can do. And one of the things they do by definition is turn into something else. So they're kind of a strange kind of biological entity right from the very beginning of the definition. And even though this definition is quite clear, the categories of stem cells in practice is actually kind of confusing and sprawling and various. This is only a partial list of the kinds of stem cells that are just part of the basic areas of stem cell biology. There's just a lot of variety among kinds of stem cell. In fact, most have nothing to do with embryos at all, for example. In other work, I've proposed a classificatory framework. It's an abstract model, but it's for our purposes, this is just a classification, a way to examine all the different kinds of stem cell and kind of figure out what are the main features that they all share. And this is some earlier work that I've done. I boiled it down to having an organismal source, a multicellular organismal source of some species and developmental stage. There's a time interval that the stem cell persists. There's some developmental process, I skipped into number 4, and then there's sort of a complex of traits in a lineage, the things that are getting compared to say that cells are more or less differentiated, for example. But stem cell biologists aren't all that concerned in practice with classifying stem cells. I thought it would be a good philosophical device to have a general framework for thinking about them abstractly and sort of capture all the variation in practice. But stem cell researchers, for the most part, are not worried about all that diversity. And I'll suggest an explanation for that a little bit later in the talk. So stem cells have a kind of dual existence. There are stem cells in artificial culture. All the little micrographs and pictures obviously are of those. And then there are stem cells in living organisms that help them develop, heal, and otherwise function healthily. And the stem cells that create organoids and embryo models are stem cells in culture. They're living experiments that stand in for stem cells in unmanipulated organisms. Cultured stem cells typically grow, they self-renew in 2 dimensions, just on flat, nutrient-rich plates of liquid media. They originate as explants from some multicellular organism at some stage of development. And human embryonic stem cells are the most famous, although not the most numerous, kind of stem cell. And so this is just a very quick overview of how they are produced. And other stem cell lines are made by variations on this basic method. So human embryonic stem cells come from very early stages of human embryonic development. And their only source are embryos donated by IVF patients. When we look at other species, stem cells can be harvested without any kind of ethical constraints, and they just grab early embryos. But in humans, the only source for human embryonic stem cell lines are a very specific set of embryos already in culture from in vitro fertilization clinics. And the process here is an embryo in culture is grown to about 5 days or so, and then cells are extracted from a part of that embryo called the inner cell mass, the ICM. A little section of that is removed, extracted, and put on a plate, artificial cell culture, kind of 2-dimensional, and very specific chemical factors are added in warm environment, and a lot of feeder cells and extra nutrients to keep the cells happy and dividing. Most of the cells don't do all that well in this environment, but a few will rapidly divide under those conditions. And when they rapidly divide, they'll form colonies that self-renew across those two-dimensional plates. And the fast-growing colonies, the rapidly self-renewing ones, are continuously selected and picked and put onto new cultural plates every few weeks. That's a lineage of self-renewing undifferentiated cells. That's a stem cell line. And crucially, that cell culture media has to include a lot of factors that actually block differentiation. The stem cells in no embryo do this undifferentiated stage persist like this. And stem cells in culture will differentiate in a lot of ways if permitted to. And that's also another whole set of experiments. But when a stem cell line is maintained, all kinds of inhibitors are added to the media. So they're kind of kept in this self-renewing stage and continue to exist as a undifferentiated line of cells. Now, when they're put into a different environment, those cells can differentiate along a number of possible developmental pathways. And we can make, this is the definition of pluripotency here, the ability to make all of the major cell types in the multicellular organism that is the source for that kind of stem cell. So whichever developmental pathway is actually taken depends on the specific culture environment that scientists provide. So other kinds of stem cell, of course, they're not all or even mostly human embryonic stem cells. Other kinds of stem cell that are cultured are produced in ways that are basically modeled on this idea. You take cells from some part of an organism at some stage of development, find culture conditions that permit them to self-renew and not differentiate any further. And then from there, they can be coaxed to differentiate along multiple, or maybe only one, developmental pathways that are possible. This is a model system for investigating development. So we also have the ability to convert or induce features like this undifferentiated cell type, pluripotency, from almost any kind of cell in practice. There's a few that are used more than others, but you can induce mature cells in culture to behave and resemble earlier embryonic stages just by manipulating the gene expression. Those are induced pluripotent stem cells. And so in practice, we can convert really almost any cell from a multicellular organism, if we have the right culture conditions, into a pluripotent stem cell that can then self-renew indefinitely in culture, and then under other culture conditions, differentiate into different major cell types, maybe all the major cell types of an adult organism. Not the full organism, though. That's totipotency, being able to create a full organism. Pluripotent stem cells cannot do that. So PSC lines, including but not limited to human embryonic stem cells, they're a really heterogeneous experimental system used to make other kinds of experimental system. And that is where organoids and embryo models actually derive from. So here's both of these are constructed by the same basic idea as a differentiation experiment. You take stem cells from their self-renewing undifferentiated line, and then put them in a very specific 2- or 3-dimensional arrangement in a specific combination of nutrients, the right biochemical and physical conditions that are calibrated to undergo development to a particular stage or of a particular organ. So the organoids are, as the name suggests, they're models of organs in a normal multicellular organism. They approximate the organs. And we have various types of brain, kidney, liver, parts of the gut, a couple of different ones, heart, retina, and pancreas. I think those are certainly the main ones, although there's probably some others. So given the right kind of environment and the right kinds of spatial arrangements to one another, stem cells differentiate a little further than pluripotency and then are seeded onto a scaffold so that they can interact to produce an approximation of one or another organ. And the standard definition, one of the most inclusive, there's a few others, but this is the one that I think has been the most influential, from Lancaster and Knoblich. The technology was innovated about a decade ago, a little bit more. So an organoid is a collection of organ-specific cell types that develops from stem cells or organ progenitors, they also fit the stem cell definition, and self-organizes through cell sorting, spatially restricted lineage commitment in a manner similar to in vivo. So I'm going to break that down a little bit more. Here's what it takes to count as an organoid according to this definition, which I think is the main one that people use. First, they're produced from stem cells that are self-organizing in an experimental context. They include multiple differentiated cell types characteristic of some specific organ. And those cell types are spatially arranged so that they resemble that organ. And they exhibit some functionality associated with the organ. So for mini brains, they'll exhibit neural activity, and gut organoids can synthesize digestive enzymes and so on and so forth. So the self-organization process is not completely understood, but we know that it involves interactions between cells and physical forces, biochemical gradients, and then interactions with the cell matrix. So it's not only things that come from gene encoding, some of that is there, but a lot of other factors as well. And that whole complex process is described as recapitulating key aspects of the way that organ develops in a normal organism. So organoids have multiple scientific uses. This is a list, 1 through 6 is from Nature Methods when they were declaring organoids the method of the year around that time. And then Science did it, I guess, a year later. So these first 6 are directly from that source, and then I added the other 3 as other things that have been collected here. So the applications that are most commonly associated in the scientific community: the access to human prenatal development and tissue maintenance, a source of tissue to study previously inaccessible phenomena of human development. The 1 and 2 are really closely related. Disease models for developmental studies, and personalized medicine and anti-cancer approaches. Those are all within the disease model orbit. Genome editing to probe the genotype-phenotype relation. Stem cell culture partners really well with CRISPR-Cas9 gene editing, and so that's another way of actually proliferating many different, very specific models. Comparative studies, number 5, bearing on primate evolution. Experimental access to the unique human brain, not specifically brain organoids. And I've added these 7 through 9 other functions and potential applications, as well as ones that are current. Drug screening, preclinical research of various sorts, and autologous organ transplantation. But the primary role of organoids, although there are many, the primary scientific role is as models in particular structure function studies of particular organs' embryonic development, especially very early stages of organ development, sometimes then specific diseases of that organ. So the idea is that by observing and manipulating these organoids, biologists can unravel those complicated interactions that are currently lumped under the stem cells or self-organizing. There's these self-cell sorting processes. Various kinds of factors involving cells, intracellular signals, physical forces, bioactive molecules, the cell matrix, and so on. And the idea is to tease apart this intricate process involving cell growth, differentiation, migration, and assembly, so as to better understand these processes specifically of human development. But of course, the models, they are simplified models. They're not identical copies of their target. They resemble their in vivo organ counterparts, but obviously not perfectly. I think the one at the top left here is a kidney organoid, retina. The second one is a retina. The mini brains are in the dish to the right. You can maybe see some of their little striations. I think the others are gut organoids on the bottom level. They don't look exactly like the real organ that they represent. And in fact, not all the cell types are included, only some usually. They're a lot smaller. And the size limit of organoids is actually one of the big areas of research right now. After about a decade or so of exploring these models, biologists who work with organoids are kind of running up against how large and complex they can be grown in culture. And the limitations actually mostly have to do with physiology, with nutrition, metabolism, and waste. After a certain size point, you need vascularization to get the cell culture system to continue functioning. And so one of the things that's missing from organoids as they've been developed in this kind of system is anything that links the different organs together into a system, vascular, immune, or neural. The components that would do that are not part of these organoids. And the limits of that kind of self-assembly are actually one of the main areas that organoid research has kind of run up against and is trying to get over. So more about that shortly. Now, embryo models. pretty similar. I'll be a little bit shorter with this section just because a lot of the method is actually, broadly speaking, quite similar. But they're different from organoids in that they are not representations or models of one organ, but of the whole embryo at a specific stage of development. So they're embryo-like structures. They go by a number of names, but I think embryo models is kind of the collection name that's being used now. They're also derived from stem cells, and they're created by adding different geometric, physical, chemical, combination thereof, constraints to the growing stem cell line. The constraints tend to be a little simpler than what you'd see with making an organoid. One of the very first of these, what's represented here is what's called an embryo-like structure that was innovated by Arya Warmflash at the Rockefeller Institute in the early 2010s. And all he did was take regular pluripotent stem cells in culture and constrain them to grow within a little geometric radius, like draw around micro patterns that constrained their growth on a plate. And when they grew to assume particular kinds of relations with one another, they started to produce this radially symmetric structure with a core of relatively undifferentiated cells surrounded by a couple of different concentric layers of more differentiated cells that very roughly, analogously resembled early stages of a developing human embryo. And so since then, a number of other different embryo models have been innovated from differently derived stem cells or combinations of stem cells extracted from early embryonic stages and produced by varying their culture conditions and, again, the geometric and physical and biochemical relations among them. So the main role scientifically of these is as models for early stages of human development. A lot of the functions and significance of what I summarized already for organoids holds for embryo models, but instead of resembling a specific organ and its early stages of development, they represent the whole embryo at some stage of development in the process. So the idea here, again, is accurate reconstruction. It's not the same as the embryo, but it's a representation of it, a model of the in vivo events of mammalian embryogenesis, particularly at the early stages when the basic body plan is laid down, many other processes as well, and when the embryo in vivo, unmanipulated, regular biological phenomena, is at most inaccessible to experimentation. So all kinds of aspects of early human embryonic development are possibly accessible, made accessible by this model system for early embryos in a way that is unprecedented in earlier research. And again, this involves sometimes multiple stem cell lines being combined, but the physical and geometric and chemical and biological properties are sort of all involved in the process. And so they're a terrific system for untangling this very complex process of development that's, in normal cases, completely inaccessible to experimental inquiry. So for most organisms, if you wanted to study early stages of an embryo, you would just collect a lot of those embryos and do that work. But of course, mammals pose particular problems for that in inaccessibility, and human embryos even more so, because there are very strict bioethical constraints on studying embryos in culture. Human embryos can be studied, but only under very specific kinds of conditions and not for very long. So the 2-week rule, I think, still holds up in practice. A lot of what we know about human developmental stages, those standard pictures of the stages, a lot of that is just extrapolated from mice, because a much more tractable traditional model organism, we don't have the ethical constraints on studying. And so much of what we know about embryonic development in humans until quite recently has just been extrapolated from other species entirely. And in fact, development is very species specific. We've learned that from stem cell research. The conditions, in fact, vary quite a lot, even among primates, let alone between mouse and human. And so the modeling role of embryo models is really has been greeted with a lot of excitement, because it offers, that's the novelty here, it offers a window onto stages of development that have been inaccessible, both for practical and for ethical reasons. But by that exact same token, because we don't have any direct access to these stages, we don't really know precisely how accurately these models will recapitulate those processes. The reason we're using them in the first place is that those processes are inaccessible. So our access to early human development, including these early stages of organ formation, so organoids are involved in this too, is solely through models like this. So there are a lot of scientific and philosophical issues that arise about these models. And so I'm not going to talk about all of them, but I wanted to kind of lay some of them out. These are some points for discussion. And again, if people are interested, there should be some time after my lecture for Q&A. So I'm happy to revisit these. Pretty obviously, one of the first things that comes to mind with embryo models is a need to really closely examine the meaning, the definition of the embryo, both for scientific and legal purposes and also philosophical purposes. They overlap. So that's one very major area that people in science and philosophy alike have been working on. Relatedly, however those definitions are settled, there's a need for new ethical guidelines that have to do with these kinds of models. There's the prospect of combining different organoids into a proto-organism. I'll talk a little more about that one later. There's distinctive ethical concerns that have been raised about neural organoids, mini brains, and they have to do with the idea of making them more realistic or keeping them in culture for longer. They're not whole embryos, so there's no constraint built into the regulatory apparatus so far. But the thought has been raised that if one was able to make more realistic brain organoids, they could develop the capacity for consciousness and experience. And in general, that's thought to be something we should avoid. There are, relatedly, a lot of concerns about public communication and trust and transparency about this kind of, all these kinds of model research, and questions about scientific metaphysics. And I don't want to suggest these are separate concerns, really. They all connect with one another. So some scientists have argued really stridently and interestingly that the way a human embryo is formed is unimportant for regulatory purposes. Looking at their history, the origins of the lineages, where the embryo comes from doesn't matter. What matters is the potential, what the cells are and what they could become. The notion of potential has been part of the debates about embryo research for decades, and embryo models really add a new layer and twist and higher stakes because the work is very easy to do, relatively speaking. We have the ability to create these things. And so it really prompts questions that are at once scientific and philosophical. A lot to think about with these models. What I'm going to do is actually not pursue most of these, although again, I'm happy to circle back to any of them in Q&A. I want to shift now to the philosophy of science dealing with models and modeling specifically. And that's the next phase of my talk. So scientific models and modeling in philosophy of science have been major topics for over 30 years, for decades. And there's been a lot of work, mountains and libraries of work on defining models, analyzing their scientific role, and examining how science looks when you think about it as primarily concerned with models. Now, defining models is obviously a really challenging thing because many different kinds of thing can be a model. And the main examples that philosophers list are equations, abstract imaginary objects, like the infinite planes, infinite populations, or perfect spheres, scale models, computer-generated visualizations, and actually simulations of all kinds, and whole organisms. But the last category, the whole organisms, is usually left out of the philosophical analyses. In general philosophy of science, the field that I primarily work in, the study is overwhelmingly on abstract models, systems of equations, or things that can be converted into a computer simulation. You can usually interconvert between those. So one of the authors who wrote the book with the Golden Gate Bridge on the cover, Michael Weisberg, he has a very inclusive account of models and modeling, and he distinguishes three categories: mathematical, computational, physical. And most of the time when philosophers do an examination or an analysis of how models work, they're really using the abstract models as their primary consideration. There's not much discussion of the physical models. Even though Weisberg has that as a category, his analysis doesn't give them any distinguishing features. He says they actually work pretty much like abstract models do. And that would be the way that if philosophers of science consider physical models at all, it's usually by assimilating them to that analysis of the abstract ones. There is a lot of really interesting work on physical scale models when philosophers of science do talk about physical models. Their classic examples are in the physical sciences. So the reason the San Francisco Bridge, the Bay Bridge is there is because there's a model, a hydraulic model of the currents in San Francisco Bay that's meant to simulate as a scale model. The little picture there is the Phillips machine. The Moniac, I think that's its name. It's a hydraulic model of England's economy from the 19th century. And then the original double helix model that Watson and Crick commissioned, I think from the Cambridge Machine Shop, would be another one of these. So like scale models are the kinds of physical models that philosophers of science consider. And again, when they analyze them, it's usually to assimilate their role to one that's very much like the way abstract models, systems of equations work. There's not a lot, as far as I'm aware, no work that argues that physical models in science have a distinctive role, that there's something specific we can do about that. Now, that being said, there's a lot of great scholarly work on model organisms, but what's interesting is it tends to be classified as another kind of scholarly contribution, not philosophy of science. There's some great work on mice, Arabidopsis, C. elegans, the book there is The Conqueror Worm, which is a wonderful title. So there's tremendous work in the history and philosophy of science looking at model organisms, and again, some really good scholarship there, and some of it by philosophers as well as by scientists. But what's so interesting is that over decades, you can see how that work, while recognized as a scholarly contribution, is left aside from the general philosophical analysis of models. It's sort of pushed into something else. It's history. It's another kind of thing. There is one exception that I'm aware of, and that's Daniel Steele, who's done some really good general philosophy of science work on model organisms. But I'll talk about him shortly later. By and large, when philosophers look at models and modeling, they just either leave living organisms aside entirely or just lump them in as a kind of physical model. And those are lumped in as basically operating exactly like abstract models. So what I'm going to argue today, and I've done in some of my other work, is that in fact, living models, generative models, have some distinctive features that are worth studying and that we can learn some really interesting philosophical things from them specifically. So I'm going to use the organoid and embryo models as a starting point to argue that philosophy of science should look at models in a different way, thinking about the living models and their generative aspects. So to do that, I'm going to focus on one aspect of the modeling, the model-target relation. There's a really strong consensus that models function in science has something to do with a target. Whatever a model is, it can't exist on its own. It has to be in relation to something else. And Evelyn Fox Keller, she's writing specifically on biology, but she has this distinction drawn really beautifully of models of and models for. So not only is it the case that you need something for the model to be of, a target, you also have to have the models for, the purposes and the interests and values of the modelers have to be part of the analysis too. You can't understand what the model's role is by thinking of it only dyadically as the model and the target. You need to have the agents and their use and their values and the purposes. And that analysis, Ron Geary's analysis there is one I'm using. It's not the only version of this, but the basic idea, and again, this is a pretty broad consensus in philosophy of science, that all of this has to be considered to understand the modeling role. You need to think about the agents using the model, the target, the purpose, and the model itself. And I think that organoids and embryo models seem like they fit this pretty well at first glance anyway, that the targets look really obvious. And in fact, when I was explaining the models in the first place, introducing them, that was pretty much how I explained them. They're models of specific organs. Each organoid kind maps perfectly neatly onto the kind of organ that it's representing. Mini brains to brains, liver organoids to livers. And for embryo models, it seems even more obvious. It's a model of the whole embryo at a particular developmental stage. There's an embryoid model called gastroloid. It's modeling the gastrula stage, et cetera, et cetera. So that would be the sort of basic approach. And what I'm going to argue is that on closer inspection, that's not the whole story. I'm not saying you can't learn anything from thinking about these models individually, but I do think to understand their scientific role, you need to start thinking about the model-target relation in a more complex way. So that's where I'm going to be going for the next chunk of the talk. Now, one complication has to do with the phylogenetic scope of an organoid's target. And the reason I'm bringing this up is that that's the one piece of model organism philosophy of science that has gotten a lot of discussion. That's the Daniel Steele research I alluded to earlier. The idea is that a model organism's role is to stand in as a surrogate for a very wide phylogenetic range, and that's their function. And so that's why the idea of phylogenetic scope is the first thing as a philosopher of science that you would look at in examining the model's role. If these are living models, you would think then they would be acting like model organisms and having a very wide phylogenetic scope of representation. That doesn't seem to be the case at all. But you could arguably think about the question being, what is the target? And for a lot of biological models, I think it's not obvious at all. There might be a very wide range here. It could be all organisms, all animals, all vertebrates, all mammals, organoids and embryo models. I think it tends to be a lot more restricted to humans and or mice. So maybe mammals, but mostly humans. So the living models in the stem cell science are generally have a much narrower phylogenetic target than they would usually be imagined to have. They don't generalize. They don't seem to be playing a generalizing role, which again is sort of the only one that philosophers of science have discussed. Sometimes I think it's not even the species. A lot of these models are actually a particular disease or even a specific genotype or members of a species that are afflicted with a particular condition. So it could be even narrower than the species range. Particular inbred mouse strains are actually, I think, another case where the species level is too coarse to really figure out what the target is. So I'm going to assume for most of what follows that the target is for organoids and embryo models both, even though the title just says organoids, but I mean both of them for now, that the target is actually restricted largely, although not entirely, to humans. So the modeling role can't just fit smoothly into what philosophers of science have theorized. And so the next obvious thing would be where I started, the idea that you just have a kind of one model, one organ, or one embryonic stage. And I do think you can learn some interesting things via that, but there are 4 features of the practice that complicate this. You can't just look at each of these models as having one target and really understand their full scientific role. So these are the 4 factors that I'm going to be zeroing in on for most of the rest of the talk. And the first is that idea of multiplexing. Why wouldn't it be enough to just think of one organoid, one target? This is an organoid issue. The idea of multiplexing organoids is the goal of combining different organoids. Again, we have different separate ones, liver, kidney, heart, brain. What if we could multiplex them, combine them into a complex of organoids that have improved function and translational relevance? This is where the limits of their growth and complexity and size have started to run up against technical innovation. There's been a plateauing of how big and complicated the individual organoid models can get. And so the next stage is to start connecting them and figuring out how to combine them with one another. And they're using biofabrication techniques to simulate or grow or engineer vascular systems that will allow the creation of more complicated organoid complexes. So the push here is to greater realism, to make a closer and closer approximation of the original organ, the in vivo tissue counterparts. So this is not all there is to organoid research, but it is a major strand of the field, aiming at these increasingly better approximations of the real thing, and eventually with the goal of plausibly producing perfect replicas that would be ripe for autologous transplants. The applications in regenerative medicine are always mentioned in this argument. And you see a similar trend in embryo research, although this is far more ethically fraught and not at all like a full steam ahead goal. It's a concern rather than this is what we should be trying to do. But the idea, and I alluded to this earlier in the discussion, is that these advances, building these embryo models and being able to construct them and grow them in culture, narrows the gap between the model and the target. And in fact, this is a really lambic quote, the model could become an embryo. You could actually erase the barrier between the model and the target. So right there, that tells you the model-target relation, something else is going on. It's more complicated than them even being definitively separated. That's one complication, the multiplexing. The second is the stem cell origin of both organoids and embryo models. So again, they're produced, I said, from another kind of model. And stem cell lines do not in any way fit a one model, one target image. They are not only powerful tools for modeling in their own right, but they are models that generate other models, including all of the organoid and embryo models that I've discussed. So I'm going to go back to make this point to look again at human embryonic stem cells. This was the experimental innovation in 1998 when Jamie Thompson's international group successfully cultured human embryonic stem cells using the method that I articulated earlier. But that innovation did not come out of nowhere. It built on a background of earlier successes culturing primate stem cell lines from other species, which in turn grew on previous successes culturing pluripotent stem cells from mice, certain other species, cancers of various sorts. You can trace the history of this kind of research as a whole set of interrelated models building on other models. And that's where the achievement came from. Months later, not even a full year, there was another publication from a different group working independently, also culturing pluripotent human stem cells taken from a slightly different place, from a slightly later human embryo, another stem cell line. And that trend has continued. Instead of there being one standard embryonic stem cell line or pluripotent stem cell line, we get a profusion of other species, other techniques, slightly different developmental abilities. And the way that the research looks is a kind of, just like stem cell lines grow and divide continuously, you get a profusion of model creation, all of which, when human embryos are involved, are accompanied by ethical discussion and regulatory work, but across other species, across other stages as well. So you get a kind of generation, not just of knowledge, but of experimental systems that are using the material abilities of stem cells directly. They're using the proliferation and the developmental abilities of stem cells to create more models that further loop back to the same process. So I think stem cell biology kind of resembles its subject matter in this way. It's got a generative quality of the cells differentiating in local environments, and the research itself has this same form as a kind of patchwork of experiments that produce an interconnected fabric of models. Organoids and embryo models are part of this family, that larger family. And they're related to one another, not just directly to their target, but again, to one another. We can understand the achievement of human embryonic stem cells in terms of the connections it has to other kinds of stem cell model. So the comparison and contrast between, not just with the target, which is directly inaccessible to us anyway, but between the different models with one another, their molecular and cellular traits, the specific experimental features, as well as the developmental abilities that they exhibit. And so you have to look pretty closely at the details of the experiments to see exactly where these ties are, where the specific connections between one kind of model and another are. But that's the way that knowledge tends to work in this field. So the thing that I want to highlight here is that the connections between models themselves are part of that modeling role. We can understand them better as a set and not just as a lump or a collection, but as an interconnected set of models. That's why I keep calling it a fabric. That's the way that this science has been aiming to understand the target phenomenon, which is mammalian or human development. So that's kind of the main sort of new philosophical thesis that I'm defending here. You've got stem cell lines or models that generate other models using stem cells defining abilities. And because of that, the models are actually physically, biologically related to one another. That's largely why they're effective. But they're not just doing it from their biological properties. The relations between models are also constructed in accordance with scientists' purposes. So it's a kind of collaboration between the cell's abilities and the scientific work that produces this intricate fabric of interrelated models. So putting these first two features together, the stem cell source and the goal of sort of multiplexing and then maybe erasing the distinction between models and targets, there's a third complication that is called looping in the model-target relation. And the idea here is just that organoids and embryo models are generated from cultured stem cells. And all of those cultured stem cells originated as parts of a multicellular organism. So you've got a continuous lineage between every organoid and embryo model to the developmental process of some organism of that same species. And on the multiplexing side, scientists are using organoids and embryo models and aspiring to create models that actually erase the distinction between the model and the target. You could eventually have replacement organs. Then you have concerns about crossing the line in embryo research. That's not a full steam ahead project. That's a concern about erasing that barrier between model and target. But that possibility is a feature of both. And so if those were realized, if the aspirations and concerns were realized, then there would be a kind of closed loop between from a multicellular organism to stem cell line to the embryo model or organoid back to the multicellular organism, an organ or an embryo. So you have generative relations that link that whole sequence between the model and the target. And the very last point is in some ways the most straightforward, that the target here is incredibly complex. Even if we limit development to humans, even if we limit it to earliest stages, it's too complicated for any single model to represent with any accuracy or detail. You need multiple models, and I don't think anyone argues otherwise. But even if you restrict this to a specific organ, you would have the same issue. Organ development itself is going to involve multiple models. Even if you were only interested in the liver, there are multiple aspects of that developmental process. From earlier and even later stages, you would need multiple models to capture it. And so instead of one organoid per organ, you would at the very least need multiple models per organoid, per organ. And same goes for all the developmental stages. Now, again, I don't think that's anything that any philosopher or scientist would argue against, but I do want to suggest there's something really specific about the connections between the models that is important as well. We don't want only to look at each model individually as it relates to its target. We should also consider how the models relate to one another. And I think the multiplexing organoids actually highlights that as well. Between the different stem cell derived models, the challenge is actually to engineer the connections between the models. And that in itself is going to be something that helps illuminate the phenomenon that we're trying to study. And similarly for embryo models, we separate out the specific stages, but the process is continuous. And so the idea that you would have a different embryo model for each individual stage of human development, the stages are all continuous and connected with one another. We want to study the connections between the different embryo models to illuminate that as well. So the image that I have, there's a really striking quote I just found just a few days ago, actually. It's the prospect, and again, I don't know that this is actually a project that's currently happening, but it's an aspiration. And the idea is actually really typical of work in this field. The idea is to have an interdisciplinary approach, not just dealing with cell and developmental biology and stem cell biology, but also systems bio, biophysics, computational modelers, data science, machine learning, and then combine the insights and methods from all of these fields so that we can learn about how to more effectively and refinedly redirect stem cells into new functional forms and use that to design an embryo model in silico, a computerized model, and then experimentally generate the digital twin in reality. So the idea of connecting not just different models, but using very different disciplines to do this, the idea that that's going to be a large-scale interdisciplinary project to accomplish. Again, this is a very far in the future, if ever, aspiration. I don't think there is a project like this happening now, although I could be wrong. This was 4 years ago. But I think it's emblematic of the idea that the connections between models and, by extension, connections between areas of science are one of the main preoccupations of this field. So to get back to my main thesis, the idea is that different models, again, it's generally agreed that multiple models are going to be needed to account for any really complicated phenomenon, but there is a tendency to think that each of those just relates to the target in a different way. And you could think of the model, the target is the sun, the heliocentric model, and every model is like a little ray, a radiation ray coming out of it. And that's really all you'd need to consider are lots of those relations. And I'm not arguing that. I think that you need to think of the models themselves as forming an interrelated fabric or network. And it's by that connected network that we actually get our understanding of the target. It's a complicated set of connections between models and their relation to the target. That's how we should think about understanding and knowledge in this field. And the source of this insight is the generative abilities of stem cells themselves. The reason that models in this field operate in this way is that stem cell lines beget more stem cell lines and other embryo constructs and organ-like constructs. We're using the stem cell's own abilities to construct these models, again, in connection with human purposes. So this is, I think, the way to think about knowledge in this field, is in terms of this interconnected set of models and targets. And there's the one goal of better understanding the incredibly complicated human development. Okay. I'm shifting to make sure that we have time. I want to make sure we have time for questions, so I'm skipping a couple of things. Okay, so to draw those threads together, and I've said this a few times, but because of these complexities 1 through 4, analyzing organoids and embryo models, just in terms of the way philosophers of science have tended to do as individual questions about one model, one target, that isn't going to be sufficient. That is, we can learn some things that way, but to really understand these models' role in science, we should think of them as an interrelated array that's constructed by a wider community, such that their relations to one another, as well as to the target, are important in making progress. So one of the corollaries of this view is that it's not a problem that we have so many different kinds of embryo model. That's a resource. That's actually how knowledge works in this field. We shouldn't be trying to standardize and narrow it down so that there's only one kind of model for each target. And I don't think stem cell biologists or researchers in this space are arguing that. But you might think that they should simplify things. But in fact, that's diversity and profusion of models is, I think, exactly how this field moves forward. It's another reason I mentioned that stem cell biologists aren't as interested in classifying their models. And I think this is largely why. They keep making more and more. And what matters are the connections between them and the way that one person, one researcher is located within this broader network is of concern, not so much how to classify as though there was some kind of static fixed way you could do that. That's me speculating a little bit. All right, so the very last point I want to make is to think about this idea of the fabric of models more abstractly. And I want to suggest, and I've suggested already, this is the form that knowledge takes in stem cell biology, and maybe in many other areas as well. It's not a single governing law or formula, or equation or set of equations that can be arranged in logical or linguistic terms. And I don't think the profusion of models that we see in stem cell research is a step on the way to that. There's no foundation and no apex. But although there's not a logical organization in the way that traditional philosophers of science have sought, there is some organization to the structure, the network structure, and it has to do with the generative properties of stem cells. We can understand the relations between these systems of models by looking at the practices of the science, which are very closely connected to and using the generative properties of the cells that they're studying. And if we get very ambitious with this is the last thing that I'll examine, is that we can think about this model-based idea, maybe not as tied to the generative abilities of stem cells, but in a more abstract way. We can think of this as an image of scientific models more generally. And so here I'm skipping a lot of steps. I have a book, the Fagan forthcoming is the book where I argue that this is a good way to think about science in general in a way that's not based on the properties of stem cells. So it looks like an incredibly hasty generalization just from one kind of model to all of science. That's not what's actually happening. But the suggestion is to think about science not as an encyclopedia of true statements that are logically organized. That's a traditional philosophical way of doing it. But think of science as a dynamic arrangement of diverse specialized communities. Epistemic communities is the term that a lot of science studies folks use, and I think it's a really good one. They have fuzzy boundaries. They're not as tidy as what I've diagrammed here, but we can distinguish the areas that science is working in pretty well operationally. And think of it as a lot of diverse specialized communities, all of which have models that represent their greater achievements, the knowledge they're producing. You can abstract away everything but those major achievements and relations among them. And what you end up with is an image of scientific understanding as a landscape of interrelated epistemic achievements, explanatory models, like what you see with stem cell research. So that pattern of relations is dynamic. Nothing is fixed. The inquiries are moving forward and new connections will emerge and fields will split or merge together. But thinking of the form of scientific knowledge in this way as a fabric of models that are connected to one another, but in a variety of ways, advancing stragglingly and unevenly, but to advance our understanding of an open-ended range of targets. And that image of science, I think, is the theme that I want to end on. There's a couple of general philosophical issues that I'll talk about all together too quickly, and then I'll stop so that we have some time for questions. The first thought is that if you think of science in this way, one question that comes up, at least for philosophers, is you think of knowledge as advancing, but in what sense could this be an advance for anyone? In what sense is it our knowledge? Any one person working in this space, in a network like this, is deeply connected to only one small part of it. And I think that's right. I think that's absolutely how we should think about the sciences, not as something that one person can deeply understand all at once, but as something that everyone occupies a certain position in. And in fact, seeing the landscape as connected, being able to locate one's own expertise in relation to other areas, that's an advance as well. That's the equivalent of making those connections between different kinds of models in the stem cell space. So in the forthcoming book, it's not out yet, but there's the cover is going to look like this. This is little themes that I'm going to be touching on. So there they are, sort of as bullet points. I call that relational understanding, the advance that an individual will get by seeing their own expertise in connection with other areas of it, thinking about that kind of integrative project that the stem cell researchers were imagining between STEM systems biologists and biophysicists and data analysts. All of those achievements can be seen as related, maybe only slightly, but you can reorient yourself as an expert and have a deeper understanding of your own expertise, seeing it in connection as part of this more complicated epistemic landscape, sort of knowing where you are in relation to others. I want to argue that that is in itself a kind of scientific progress, very connected to more traditional notions of knowledge. And if you think about it this way, if you have this image of science and sort of follow it through to its conclusions, it has some very obvious applications to interdisciplinary research. You can use this idea to scaffold further interdisciplinary connections and projects. I have an account of how to make minimal connections between very different explanatory models. And if you can do that, you can then use that to feed back and generate further common ground and shared values. And so the more that science uses this image and works in this way, you would have a mechanism for getting a gradual accretion of a more inclusive scientific community through interdisciplinary activities that would advance aspects of shared understanding. And that is the version of unity of science, what I call patchwork unity of science that I'm articulating. And that is a very big departure from the traditional view. This is not a current piece of philosophy of science, but it is an influential background idea from the mid-20th century, really influential work by Oppenheim and Putnam and their colleagues, that science forms a nice orderly structure, hierarchically arranged through levels of generality. So the most general sciences that have laws of greater generality that subsume those of less, and you can build from a foundation of the most secure, the most general accounts of explanatory of elementary particles, all the way up to accounts of social groups. But this way of viewing the sciences, again, has a very obvious ordering of what the most fundamental and the most explanatory sciences are. And although I don't think too many philosophers of science today view science in this way, the idea that there is this hierarchy of the sciences, that here's this is an ontology structure, but Oppenheim and Putnam argued that that actually corresponds to the branches of science. Branches of science line up against these levels of reality, and you can articulate the hierarchical relations among them, and thus capture the form of scientific knowledge in this way. And so what I'm proposing is something much more confusing and messy and not hierarchical in the slightest. But I think it's actually a more accurate and fruitful way of thinking about scientific knowledge in our current context. So again, I'm going way longer than all right. I'm going to say one more thing and then I'll pause. I'll just want to make the last 2 points here. Besides it being anti-hierarchical, the way to think about some advances in science here are just making those connections. And in fact, thinking about explanatory achievements as even within a community like stem cell research, they're a collaborative effort as well. And if you think of science in that way, there's an epistemic advantage. You can actually advance knowledge by using pro-social attitudes. Of course, that's not all there is to science, but there's been a lot of emphasis in science studies on competition, groups competing to get the right answer. I think it's not that that isn't the case, that there's no competition in science, but there's also the pro-social collaborative aspect that you especially see in interdisciplinary areas. And so the norms that come from thinking about science this way, like the constraints for how to do science, how to get better explanations, how to get better understanding, those are exactly the same things that get better collaboration, attitudes that are associated with that. And that'll be the last thing, the last point that I'll make is that if you think of science in this way, as a collaborative network of very loosely connected communities, then science-society relations do not look the way they do traditionally as well either. There's no demarcation between science and society because there's no one thing that's science. You can certainly draw boundaries between different communities, but there's no absolute sort of wall around the sciences. And in fact, the opportunities for seeing connections between scientific and other kinds of projects are just multitudinous in this sort of image of science. So that's the last point that I will end on, just so that we have time for questions. Thank you so much for your attention. There's my supporters and the norms in science and philosophy about Q&A are completely different. So I don't know how this will work.
I do have a question, but I didn't know if there was any in the chat.
Not yet.
Okay, so with your network image, this one. I'm wondering if you could speak to, so all these are kind of within the realm of biology and health science.
This one, yeah, I didn't want to try to do it.
No, I get it.
And actually science, I mean, obviously some of the big data sociology folks have done that. Like there are
Right.
bibliometric designs primarily, but not only, and there are models, but they just look like, what you would imagine they look
But I
like.
think about this idea, right, the relationality, especially between science and society. I think this is kind of an easy one to think about because of health science being so ingrained in science and in society.
Yes.
How do you think this might work if you were to think about like some very bottom of the tier elementary particle physicists and their interplay with society?
Awesome. So the first thing I think, and again, that's outside my own, there's a reason it's the life sciences.
Okay.
There's my disclaimer, but there's also, I'm going to go back to some of the really groundbreaking work in sociology of science used exactly those high energy physics, examinations of like how the technology doesn't come out of the ether. And the ideas, neither do the ideas. There's the kind of very traditional abstract philosophical history of the progression of physical theory, but I think a more well-rounded notion of the history of those insights also connects with technologies. And as soon as you're doing that and you bring in those dimensions, then the questions about resource allocation and the way that the site, like the choosing of the site and the way it interfaces with, in fact, questions about labor and who is doing the scientific work. I actually think that shows up in really interesting ways in biology too, like the hands doing the work are a really different class than the people who are getting all the credit quite often. And that you see a version of that in the hard sciences as well. I mean, again, and I think it's going to play out differently. I'm a particularist in how to think about the organization of science, but I think any field that's in, sometimes the hard sciences or any science is going to be doing work that's sort of insular. And there isn't. So I don't want to suggest that everything is going to be connected to society, but I do think that any field that's like building anything and constructing anything. There's going to be some way in which it interfaces with other projects. So the ones that I'm interested in, there's a much broader scope for inquiry here. But the parts that I'm interested in are when people are investigating something aimed at understanding. And so not every other social group is going to care about that. There's other kinds of concerns, and it might be that you need to specify the difference between that and something else. But often when there is a social group that's concerned with understanding, then there's going to be some way that you can connect the achievements that they have in mind. And so I think actually extending the idea of interdisciplinarity so that it applies to science and society relations. So I think the prospects for that, they won't cover all aspects of the knowledge. I think it's pretty unlikely that some of the most technical and specialized things in any field are going to interface directly with the outside, but that's okay. That's, oh, did I lose them?
There you go.
Okay, sorry. Anyway, so that, and it actually just has to, a lot of the work for some areas of physical science has already been done and hasn't been really taken advantage of in the philosophical studies of this. The philosophical studies that exist of models and modeling and explanation, I think, are great, but they've been using a very tiny slice of activities as all that they're using. And if we broaden it out, there's actually already a lot of work that we could see, like the library of work that's relevant to getting insights here is actually much more extensive than the literature so far would suggest. So it's really like thinking about what's relevant to thinking about explanation. But earth sciences are another place, like the ways that exact sciences and like hard physical sciences connect anywhere, anything in the relations of technology development too, like all of those things are going to be places where you'd see this. I see a couple of hands.
So what way you might be able to test this model and like you've done this, take any one person's publication and then do this kind of system modeling with it in order to see how that goes out in what fields and that with those citations.
Oh, the separate, yeah. And the and again, I think it's been a while since I looked closely at, but informatics has a lot of, there's lots of data crunching abilities. So there's some representations, I doubt it's of everything, but I think that kind of interconnection, then you get a network with properties that you can investigate, right? Is that the thought? Great. I'm more interested in the closer connections because I think that while you will be able to do, and I think people are doing that kind of analysis with the bibliometrics, just because that's the easiest or one of the most accessible forms of data for this. I guess what I'm more narrowly focused on is how different fields achievements, like the specific models that they construct, could be seen in relation to one another. And in fact, so the random selection of any one-to-one, that probably wouldn't be informative. Most of the time, there wouldn't probably be any need to directly connect those models. The way that I articulate it is there an interdisciplinary project where they're trying to? And then when you're trying to, then you can start, there's a motivation there somewhere, there's going to be some reason to start making those connections. So the kinds of connections that I'm diagramming in here are not going to directly map onto a bibliometric maps, but it would be super interesting to look at the one kind of network representation and the one that I have, and then see where their different properties are. That would be fantastic. That would be really fun to do. Thank you. Yes.
Yes, I'm sympathetic to most of this. I'm a fan of Cartwright, Dupre.
Oh, so am I. You might have recognized the influences then.
Yeah. Where I'm a little bit worried, where someone else might be worried, is
Oh, you should. People should be aware.
we're really just thinking about models and the distinction between models being identified as opposed to models being built and constructed. So thinking about, like simulations and so on, climate stuff.
Oh,
so
yeah,
like, Winsberg's work, thinking about how we can build up these simulations to hone in on learning something very particular of interest.
Yes.
So it has a kind of pragmatic constraint. But whereas in treating these like biological phenomena as organisms, they seem pretty constrained just on their intrinsic interests. So I'm curious why still use the language model there, not something like causal antecedent or something like that. is potential of like.
Oh, yeah, yeah, great, great. So this is, oh, go in the wrong direction. Yeah, so, where we go? Oh yeah, that's the one, that's the one. So I've drawn these as directed just because I was looking at in this left-hand thing, the time order of when they were created. But the reason I call them models is that I think we're using them to understand the target better. And so when I use models, it's actually in a little bit more relaxed sense that a lot of the philosophers of science would. They will sometimes have a restriction. Some of the definitions wouldn't even allow these. They would just call these direct experiments. Weisberg and PG and Godfrey Smith would say that. But that's because they're using the term model in a more restricted way than I am. So I didn't put that part of the argument in, but you're absolutely right. Like some philosophers have a notion of model where it would actually look inappropriate that I'm using those terms. But that might not have been the point you were making, but what I would argue is that the term makes sense because we're using them to understand the target. That being said, there are very clear and profound differetions between models in climate science and models in this field, and the particularities of the way that they're being used. I think we should absolutely not lose sight of that. I'm not aware of any direct connections that are being sought between climate models and these kinds of models. It's hard to imagine a project where that would happen, but there are a lot of connections between climate models and other kinds of models. And actually an early version of my view was applied in that sphere, although it's by Monica Morrison. And so that would be where to look. I wouldn't try to make the argument that there should be a direct like network connection that's advancing understanding between climate models and these. But I do think some other fields, modeling achievements, are going to productively need to make those connections. And that's actually how we should understand climate models, not as like this thing's totally unique in science and unlike other things, but things we can productively link to other projects. That would be the way I would articulate. I don't know, did that help
You know, I think that's good. It's just
answer your
that,
question?
like lingering worry would be then could potentially everything be involved.
Yeah, that is a worry, but I'm not as worried about it as a theorist about models because I'm trying to talk about explanation and understanding. So that's the privilege. I'm like, yeah, the people who are theorizing models are going to use it in a more specific way. But you're absolutely right. I think so. Oh yeah.
So I've been interested in the interdisciplinary stuff for a long time, but I've run into like kind of barriers just because of the institutions already being set up. If you were controller of the universe, or just if you had any like individual advice or any systems that you could see allowing those interdisciplinary connections to grow.
Goodness. No, it's so awful. The first thing I thought was a negative thing. So I'm going to think a little bit more, but because we only have a couple of moments, maybe it's positive. My first thought, one of the things that I've been thinking about is how to best use your energies and when to give up on a connection. So this is the first thought, is that sometimes one of the first things I try to look at are what gives some sort of minimal constraints to making a connection between models. And if you can't meet those, then it's like, great, I will work around this. I'm going to find another. So it means you're at least not wasting time trying to force a connection where there isn't going to be one present. That isn't the answer that you wanted. Then I'll think a little bit harder and see if I can come up with something over a little more time. But I do have a lot of thoughts about this issue. It was one of these, like, which of these comes out first and in 1 minute. But I guess the positive side is actually centering the pro-social values. And that's the answer to the negative question too. It's like actually seeing those attitudes that go into being a successful collaborator as central to scientific practices, not just something you would do like, oh yeah, if you want like an extra little credit on your name, like here's an interdisciplinary thing, but it's actually central to explanation and understanding. And so having that be, if I were able to control like what people said or thought, I would center that more as the image people have of science, and then try to think about how we can best support that institutionally.
All right, I think it's that time, so we better get going.
Thanks so much. I hit the.
This was like, it was slowly moving, like it's got tension on it. I was like watching it disappear slowly.
Did I? Okay, good.
Perfect, we can-
And that, I think it's muted now, right? Oh, yeah.